We'll now move to a panel session on using brain cell atlases for brain disorder research. This panel will focus on how knowledge from a brain cell atlas can enable biological discovery from human genetics and clinical research, how it can generate testable hypotheses about pathophysiological mechanisms, and how it can facilitate collaborative research efforts. Our two moderators for this panel session are Drs. Evan Macosko and Nenad Sestan. I've already introduced Evan, so I'd also like to introduce Nenad. He's a Harvey and Kate Cushing Professor of Neuroscience and Professor of Comparative Medicine, of Genetics, and of Psychiatry at Yale University School of Medicine. He studies brain development and evolutionary relationships of brain development across species, particularly humans, non-human primates, and rodents. Evan and Nenad, hand things over to you for the panel session.
Can you hear me?
Yes.
Okay. So good morning, and okay. So can you all see the slides?
Yes.
Okay. You can still see them?
Yes.
Okay. So good morning and good afternoon, everyone. My name is Nenad Sestan, and together with Evan Macosko, I will be moderating the first panel of day 3 of this workshop. I will start by thanking David Panchisian for the introduction and his NIH colleagues for their support and many wonderful contributions they made for all of us. So thank you. I would also like to acknowledge Steve McCarroll, Eva Macosko, and Philippe Dayeger for their outstanding keynote presentation and pretty much basically a wonderful introduction into our panel. I think that they have covered most of the key points that we'll hopefully be able to discuss. And so our panel today will discuss the use of brain atlases and cell atlases for brain disorder research. I would also like to thank our note takers, Xiaojin Chen, Mike Haverlich, and Carol Thompson, as well as our panelists listed here on this slide. I will now turn things over to Evan to summarize the two main themes and key questions. These are not the only questions we are limited to that we would like to address in this panel, and introduce our speakers, Genevieve Konopka, Kimberly Siletti, Mercedes Paredes, and Jason Stein. Evan, I can forward the slides.
Yeah, sure. Thank you, Nenad. And we want to keep this introduction very short because, as you can see, we have a very distinguished and varied group of panelists who will be joining us, and we want to make sure their contributions to this area are really they're able to voice them to the group. And we also have some really fantastic speakers who are going to kind of set the stage for us on these two major areas of questions. The first sort of centers on the overall opportunities associated with Atlas data and learning kind of basic understanding of human neurobiology, and especially with relationship to what distinguishes the human brain from the brains of other species. And our second theme will be focused much more on kind of pathophysiology and disease mechanisms that we can learn. So I think what I'll do is I will just introduce all four. Each of the four speakers are going to have prepared a short presentation for us to kind of outline their and sort of highlight some of the themes that we'd like to bring up in this discussion. I think what I'll do is I will introduce all four of them now, and then kind of go to each of them individually to start their presentation. So to begin with, if you go back to the previous slide, Nenad, we're going to start with presentations from Genevieve Konopka from UCLA, who will tell us about her use of single-cell data to understand molecular features that are driving human-specific brain cell complexity. After that, Kimberly Siletti from UMC Utrecht, who built one of the first human brain cell atlases with single-cell and single-nucleus RNA-seq, will tell us about that work and insights from more recent cross-species analyses. And then in our more focused discussion of the pathogenic mechanisms, Jason Stein from UNC will be telling us about efforts to assess the fidelity of human organoid models and how they are working to test gene by environment interactions within organoid model systems. And finally, Mercedes Paredes from UCSF will take us on a very focused and very fascinating study of an epilepsy case that was studied using single nucleus RNA-seq. How about we'll start with Kimberly.
So yeah, as mentioned, I'm going to talk a little bit about sort of the atlasing work we did, some recent cross-species analysis. So it's along the theme of human neurobiology, linking to disease a bit, and in particular, trying to bring some of the focus to incorporating the whole brain into these types of analyses, or doing these types of analyses across the whole brain. So as was mentioned, I worked on the version 1 of the Human Brain Atlas, which was I think as everyone knows, a collaboration with the Allen Institute. In particular, Rebecca Hodge there coordinated over 100 dissections from across the whole brain. And although this was heavy on dissections from the cortex, as you can see in the schematic, we also sampled the other structures as well. And so this includes hypothalamus, thalamus, and parts of the brainstem as well. And one of the most interesting challenges or questions that came out of the data for me was how to make sense of the astonishing diversity that we found outside the cortex. So we used an iterative approach to clustering the data. And at the finest level, we found over 3,000 subclusters. And a third of this diversity fell into a single supercluster of neurons, which you can see looks quite complex and overwhelming to make sense of. And this supercluster of neurons came from almost exclusively outside the telencephalon, so outside the cortex and hippocampus, so largely the hypothalamus, midbrain, pons, and medulla. And this was surprising because we had not sampled these regions as deeply relative to the cortex, which suggests that these brain regions are made up of really very, very many lots of rare different types of cells, which now has also been seen in the mouse as well. And one of the other challenges about this data, which has come up a couple days ago as well, is that this diversity is also difficult to make sense of because it's not so clearly hierarchical, which I just wanted to take a moment to illustrate here. On the left-hand side, so this is the dendrogram of the whole brain you're looking at, and on the left-hand side, you can see these excitatory and inhibitory neurons in the telencephalon form these two distinct types with nice distinct subgroups, which have even further subgroups under them. But when you go to the rest of the brain over here, you can see that this structure really starts to fall apart, and you start to see a much more flat looking structure, and excitatory and inhibitory neurons mixed together. So I became very interested in what generates this astonishing diversity in regions outside the cortex, like the midbrain, for example. And I started thinking that this diversity must be very highly conserved. We also see such high diversity in the mouse. These regions are evolutionarily very conserved. So I started thinking about ways that we could test this experimentally, and I was really interested in the midbrain in particular because you see these two different types in the midbrain. So you see that many of the neurons fall into this splatter neuron type, this really highly complex supercluster I mentioned, but many also fall into this midbrain-derived inhibitory supercluster here, which you can see looks quite different. It's a relatively homogeneous cell type relative to the other midbrain types. And this supercluster was named midbrain-derived inhibitory neurons because it was a supercluster that contained exclusively midbrain and thalamus almost. And there had been papers describing that neurons migrate from the midbrain into the thalamus during development to populate the thalamus as inhibitory neurons. And so this seemed like a very reasonable naming for this supercluster. And so I thought that we could try to pick apart the origins of these two different types. So we just needed to go to the mouse and identify which cell type these were. So we did a cross-species comparison of a human and mouse atlas, and sort of no matter what configuration we come up with, we came across this one large group of human neurons that didn't really mix well with the mouse. And you guessed it, these were the midbrain-derived inhibitory neurons. And now this makes a lot of sense in light of work that's come out of Van Akrenen's group in the last few months, I think. they found that these thalamic inhibitory neurons that are so enriched in primates are largely midbrain derived. And so that's what we're probably seeing when we're doing this analysis. This supercluster represents an expansion of midbrain derived inhibitory neurons, likely in fact. And there are now, I see that this naming is actually somewhat misleading. There are probably multiple types of midbrain derived inhibitory neurons, some of which is primate innovation. And you can get a hint of that here. So actually, if you look back at this very complex supercluster of neurons, there are clearly clusters where thalamus and midbrain also overlap, suggesting that these are also a similar type of neuron, perhaps one more conserved. And we also see in line with the developmental data from Tom Nowakowski's group, we see that some of the thalamus neurons align with the neurons in the forebrain, suggesting that they have forebrain origins instead. And just to emphasize that there are even further challenges here, when we're looking at these particular neurons across species, we found really nice markers that looked really beautiful in the human. DMBX1 is the major example. So you can see it's very nice, clear marker here. And then what's so strange about it is it's a really beautiful marker in the mouse as well, but not for the most closely related mouse subtype. So there's a lot to pick apart here in making sense of how this expansion might have occurred. And I just want to wrap up by emphasizing that this human neurobiology is also critical for disease. So I'm part of the Brainscapes Consortium here in the Netherlands, which is a consortium that aims to link cell types to disease. And my colleagues performed a single-cell nucleus single-cell RNA sequencing atlas of the human ventral tegmental area in the midbrain. And in their analysis, they also find these, what I called midbrain-derived inhibitory neurons. And they see that these are enriched for genes that are involved in psychiatric disease. So this is clearly a very exciting time because we now have this new insight into human biology that could really start to crack open these diseases. This is a major new cell type, for example. But we also have a lot of challenges ahead of us because now we are seeing that we're going to need new models. And so that's all. I just want to quickly wrap up by acknowledging everybody who was involved in the work that I had on the slides here. And yeah, thanks again for the invitation.
We do have a question from Arnold Kriegstein if there's time.
Okay, yeah.
Thanks. Very interesting, terrific talk. I was curious, Kimberly, when you mentioned these midbrain-derived inhibitory neurons, have there been developmental studies looking at an actual origin of this cell type or any of these other midbrain cells? Do they actually come from progenitors in the midbrain earlier on?
Yeah, so at least when I was annotating this data, there aren't so many studies, but there, I think, was at least one study in the mouse which had done lineage tracing using a midbrain marker and found that these thalamus interneurons derived from the midbrain developmentally, which informed my annotation.
Yeah, very interesting. Thank you.
I think that's an excellent question, but how, that would need to be done in primates.
Yeah, it's a real challenge, I think. And yeah, I think primates is an excellent opportunity to really try to understand what these do. Yeah. And then maybe there are other platforms as well for maybe linking some of these to disease variants and things like this, but so somewhat multimodal. But I think there's a lot of promise in the primates. Yeah.
Tom, you have a question?
Oh yeah, thanks. While we're waiting for the slides, maybe I can just weigh in. So in human developing tissue, you can see indeed those midbrain derived interneurons that are actually locally proliferating, or there is evidence of intermediate progenitor cells that still bear markers of the midbrain lineages, including IRX3 and other classical markers of the midbrain, but they seem to, in humans at least, locally proliferate in the thalamus. So there could be a situation where a progenitor cell is derived from a midbrain and then locally proliferates to expand the population. So that's at least our current working hypothesis there.
Jenna, I have your slides, so if you want me, I can share them.
Yeah, so I only have a few minutes, and today I'm going to focus on our previous work and where we're moving, and coming on the third day of the workshop. I think you've heard a lot of kind of the take-home messages I want to say, but I'm going to really be focusing on brain cell proportions and how we can look at them specifically in humans. So next slide, please. Yeah, so first, I just want to thank the folks involved in my team that are part of my BICAN R01 as part of the atlasing effort. So Stefano Berto at MUSC, Bill Hopkins, MD Anderson, Chet Sherwood, George Washington University, and Sujan Yi at UC Santa Barbara. And this really takes a team effort to, as many of you know, as you've seen in all the acknowledgement slides, to do these atlasing efforts. So next slide. So my group and all of the groups that I'm working with, we're really trying to understand if there are any features of the human brain specifically that we can uncover at the genetic and molecular level. And we think to do this that we need to be comparing closely related species such as other non-human primate, great apes, ideally chimpanzees. So a lot of our work has been using a lot of closely genetically related species. Next slide, please. And so in this type of approach, we're typically comparing, in this case, a human compared to, say, chimpanzee, and then using as an outgroup another primate species, typically rhesus macaque, although you see in one slide and in some of the unpublished work of our group that we're also now, and as part of the BICAN atlasing efforts, using data also from marmoset. And so just click one or two more times, please, Nenad, and one more time. Yeah, thanks. So in this case, we can identify, go back, things that are changing specifically on the human lineage. And what I want to emphasize here is what we've been trying to do moving forward from our work from over a decade ago, where we were looking in bulk and using cell sorting approaches, is really to take a cell type approach and not just to focus on neurons or other cells that we've heard about in this workshop, like microglia or astrocytes, but all the cells, and to not be biased in our approach to neither sort, but to really focus on everything that we can find. And there's quite a diversity, as you've seen over the past few days. And so we were really primed based on our previous work when we were comparing human chimpanzees and rhesus macaque using these microfluidic approaches to look at the oligodendrocyte lineage. We had some inkling something was changing specifically in oligos. And so when we had this study looking in the posterior cingulate cortex, if you could just click 2 more times to show the rest of the slide, what we found was that when we could really subset all of the different cell types on the oligodendrocyte lineage, was that there was this very striking change in the proportion of cell types. So, we're all very focused on looking at differential expression, but these data, looking in this unbiased manner, suggested to us this increased proportion of immature oligodendrocytes, specifically in the human lineage, and a decrease in the relative proportion of mature oligodendrocytes. And on the right, what you can hopefully see is when we quantified this using both the single nuclear RNA-seq and ATAC-seq, we saw this increase, statistically significant increase in the immature oligos and a decrease in the mature oligodendrocytes. Next slide, please. But as many of us know, to do these microfluidic approaches, we have to dissociate the tissue. And so we were concerned that perhaps we were biasing our results in some way to see this proportional change by either by the dissections or what survived. And so what we decided to do, and as you've seen over and over again, and spatial transcriptomics will kind of fix this, was that if we could take the tissue intact and look, we wanted to know if we could recapitulate this. And so we did some RNA scope experiments in independent tissue samples from human and chimpanzee, and we could recapitulate this finding of increased immature oligodendrocytes as indicated here by the PDGFRA signal, and a decrease in the mature oligodendrocyte signal using MOG. So this is really reassuring to us. Next slide, please. And so what we were able to do using the data that we generated and publicly available data at the time was to look to see how this change in proportion held across other areas of the brain. And so everywhere we looked in the cortex, as indicated here in these 4 neocortical regions, and now including data from marmoset, we could see this alteration of increased immature, decreased mature oligodendrocytes. And in the 2 regions at the time that we could look at that were non-neocortical, we didn't see this proportional change. And so I'm very excited about all the atlasing efforts coming out, where we're going to be able to look across the whole brain, across many of these, if not all of these, well, not chimpanzee, but in many of the species, to see how these proportional differences do hold up in other cortical regions and whether there are changes in non-neocortical regions. Next slide, please. And then the other thing, if you can just click through this slide, I think there's 2 or 3 animations that we were able to do was, all of these data sets are from postmortem tissues, and we don't understand, that's good, what the implications are for this change in proportion. And it's, ethically impossible to study this in a human in a lot of these comparisons. And so now, as we've seen in other days of the workshop, what we can do is take iPSC-derived cells, in this case, human and chimpanzees, and study this function. And so my lab has developed a new, faster way of differentiating iPSCs across the whole oligodendrocyte lineage with these cells alone. And we were able to recapitulate, using comparing human and chimp, this change in proportion. So able to see a decrease in mature oligodendrocytes as indicated by mature markers and an increase in immature oligodendrocytes. So now we can study functional outcomes of this effect and other atlasing observations, proportional or not. And we can also manipulate by testing contributions of specific genes. And we could co-culture the oligodendrocytes with neurons or other cells and see the functional outcome of this change in proportion. Next slide, please. And so what is missing? So how can this sort of approach contribute to Atlas effort? And you can just click through the text, please. Well, what we want to do is, for example, focusing on the oligodendrocytes, our grant is really to do a systematic analysis of white matter tracks, because we've all, as a field, really been focusing in gray matter and neurons, and do a regional comparison of white matter tracks. Are all oligos and their precursors the same transcriptomically if you look throughout the brain? And how do the proportion and gene expressions in oligos change over development? And one more click, I think it says, and how does it change across and between species? And so we're comparing overdevelopment with our Atlas efforts, all of these white matter tracks, and across all of the model species that are included in the Atlas efforts. Next slide, please. And so take-home messages, and you can just click here: treat all cell types equally, don't have a bias, think about and consider these proportional differences across regions, development, species, use spatial transcriptomics. We'll probably fix a lot of these issues moving forward, and these independent validation experiments, including the RNA-scope and iPS-derived cells. And that's all I have to say in the slides. I just have a thank you slide at the end, and I want to thank Nenad for clicking through, and I apologize for the technical issues. Thank you.
Thank you so much. Let's move on to Mercedes.
Well, thank you to Yang, Evan, and Nenad for this invitation. My name is Mercedes, and I will be discussing this ongoing kind of shift into wanting to not just think about the neurotypical brain, but also using our emerging technology to understand brain disease states. I think the keynotes, especially from today, really highlighted the power and the potential for understanding mechanisms. And I also want to think through a short vignette, thinking about how we can use this in real time, potentially patient care. And to do this, I want to think about and focus on the applications of spatial transcriptomics. As a Californian, I think a lot about heterogeneity of terrain, and the brain, I think, is very similar in terms of we want to think about how do we apply all this beautiful cell annotations and give the brain, which is a very diverse, as you all heard through the many talks throughout this workshop, diverse space, precise spatial labeling, and how do we give spatial resolution to the cell type specific gene expression that's starting to emerge. And I think we've heard some very amazing examples. And today I'm just going to share a short vignette really led by an amazing MSTP, a student, now currently a clinical fellow, Konopka, and trying to apply this technology to neurodevelopmental conditions. You've heard a lot today about neurodegenerative conditions, and I'd like to think about this also in another setting, specifically in epilepsy. And in particular, we became fascinated by one case report of a condition called hyaline protoplasmic astrocytopathy. This is a pathological finding that classically is seen in brain tissue resections, where you see these eosinophilic and refractile cytoplasmic inclusions, specifically in cortical astrocytes. And these are typically seen in resections from epilepsy cases. And that's been an increasing interest in terms of what is the role and function potentially of these inclusions that have been so classically a pathological feature. And increasingly, analyses of these HPA cases have been associated with malformations of cortical development, also known as MCDs, such as focal cortical dysplasia. Those are conditions of disrupted brain development that have been associated with very refractory and medically resistant epilepsies that are often treated by surgical resection, thus allowing us to gain access to tissue from these patients in real time. Because of these access to tissues, increased genomic analysis has now associated the presence specifically of a 1q gain microdeletion with these HPA cases. And interestingly, these are not often germline mutations, but specifically somatic mutations that can only be identified by interrogation of the brain tissue. And so again, there's been a lot of interest in what are these inclusions that are such a classical feature in neuropathology? Do they have a mechanistic insights? And what does this give us in terms of understanding the pathology of HPA cases that I think are increasingly being recognized? And how does this influence our perspective of mechanism? So with one of the cases, Kung took these cases and performed single nuke RNA sequencing and identified the cell types, and particularly then created what he called a 1q module scoring, where he looked at the expression of genes on chromosome 1q and compared that to the expression of all other genes to create this module score. And when he did that, he saw that there was an increased score specifically in astrocytes and in neurons that was identified. Furthermore, when he looked at astrocyte population, given the cases showed pathological findings in astrocytes, he particularly noted that a subpopulation of astrocytes were enriched in synapse-associated transcripts, genes like RIN1A, as well as others. And so that was interesting and suggested why would these astrocytes have synaptic genes present, particularly notable in cells potentially that would have these inclusions. And interestingly, our study and the findings came out when the Aaron Heisens and Tracy Bedrosian's groups also published a small cohort of 5 individuals with somatic mutations with a 1q gain. And also they had identified in their single nuke assays this 1q gain present specifically in astrocytes, as well as the enrichment of synaptic genes in their astrocyte population. But how do we connect the presence of this 1q gain and potentially synaptic genes to the inclusions observed pathologically in HPA? So to tackle this, Kung, an amazing senior scientist in the lab, Julia Chu, turned to spatial transcriptomics to actually interrogate and localize these inclusions within the tissue and try to tie those with their transcriptomic signatures. To use this, they applied the Xenium 10x platform. And this thought was here that not only could we look and interrogate their transcriptomic profiles, but also combine that with both immunohistochemistry as well as neuropathological immunostains, such as NISL, that are so canonical and necessary to identify the pathological features. I think this also kind of highlights kind of the economic aspect that potentially could be applied clinically for use of these the strategy, looking at multiple sections. And here we could see that we could look at 6+ individuals on a single slide and assess them and actually obtain quite a robust cell number consistently across samples, increasing our potential power and the potential ability to do many case analyses. So taking over a dozen cases, and these were cases not only from our own inventory at UCSF through the collaborative efforts of neurosurgeon Curtis Augusti, as well as neuropathologist and physician scientist Catherine Cadwell, but also from Tracy Bedrosian and Aaron Heisen's own cohorts of 1Q patients, and as well as other 1Q patients generously shared with us by the Magaki lab at UCLA. And with this cohort, we were able to see that the Xenium data alone could give us representative cellular populations that we had observed in our single nuke assays as well. Doing that, we could also then start to tie some of the neuropathology features, again, the presence of those inclusions with the transcriptomic profiling. Here you can see the segmentation of the hilum material in pink, highlighting those inclusions, and then you can overlay that pathological stain with the cell annotations shown here in teal cyan for the glutamatergic neurons and in yellow for astrocytes. And so then we can actually identify the presence of the 1Q gain in specific cell types and associate that with the pathological signature. In doing that, we were able to perform spatial classification of their astrocytes in our 1Q cases. We could see that our cluster 1, when we subclustered our astrocytes, cluster 1 was really associated with the gray matter astrocytes, and cluster 7 associated with white matter astrocytes. performing a 1Q module score. So here we could use a segmented masking to identify the inclusions, create a module score for that. We could see that the hyaline inclusions really were localized to the gray matter astrocytes, as well as a subset of neurons in our HPA cases. We could also see that the inclusion-specific gray matter astrocytes we identified also had a higher chromosome 1Q module score, really suggesting that the robust presence of this 1Q was starting to focus in this inclusion-positive astrocyte population. Lastly, we could see that within the inclusion-positive gray matter astrocytes, there was a diverse set of upregulated genes, some that were present on that 1Q arm, chromosome 1Q arm, and associated with potentially abnormal astrocyte functions. For example, the astrocyte-specific sodium-potassium channel, as well as GLUT-A. But we also were able to identify genes that were upregulated that were not present in the 1Q arm. Genes like TNC that could be associated with kind of overreactive astrocytes, as well as synaptic-related genes such as SNAP25A. Again, highlighting the presence of unusual neuronal genes in these astrocyte populations, even if they were not themselves disrupted in the 1Q arm. So right now, this leaves us with this hypothesis that the 1Q gain, particularly in astrocytes, could lead to abnormal function of these astrocytes. For example, leading to abnormal phagocytosis of neurons, as suggested by the abnormal presence of synaptic genes within these astrocytes. This led us to think that maybe potentially we could study these in vitro and start to assess the phagocytic capabilities of astrocytes with the presence of the 1Q gain. Our spatial transcriptomics, I won't get to show you today, has also kind of collaborated this by the spatial relationship of these inclusion-positive astrocytes to adjacent inclusion-positive glutamatergic excitatory neurons. And so I want to end today with thinking about how spatial transcriptomics can really lead to high-yield information that's really giving new insights and discovery to human neuropathology. We can look at this in the targeted genes, such as using a targeted panel in spatial transcriptomics, but also looking at disease relevance by directly linking cell types with clinical, potentially diagnostic markers, now that we would have the ability to combine our omics with classic neuropathology assays. I think as we continue to enhance the modalities that are being used in these multi-omic approaches, I think that'll only increase our ability to interpret and take these platforms both from the bench, potentially into a clinical setting. I think one thing I would love to continue to think about is how these platforms can be tailored for the quality and processing of the diverse tissue that we're all trying to use when looking at the human brain. We heard a lot about postmortem samples. I shared with you the kind of the reproducibility and the robustness of using actual neurosurgical samples. But each of these samples and tissues really behave differently. We have to consider that as we think about the applied platforms that we're going to be used for each of these conditions. I want to thank you for the attention. I want to thank the team, and in particular Kung Loon, who drove this project with collaborators Catherine Cadwell and Aaron Titus and Curtis Augusti, as well as collaborators who shared their cases. And I think it's really exciting to think that we can start to look at diverse cases, both from new and current samples, but also from old archive samples with the robustness of the technologies that have been emerging to analyze these tissues. Thank you so much.
Thank you. And lastly, let's turn to Jason.
Okay, so brain organoids are a unique tool that allow us to study human brain development. They involve the acquisition of cells from an individual, reprogramming back to a pluripotent state, sort of going back in time, followed by differentiation of the quote-unquote organoids to model the processes of neurogenesis and maturation. And importantly, they can be derived from individuals with psychiatric or neurological disorders, to try to understand the cellular and molecular mechanisms that lead to those disorders. Organoids, though, are a ball of cells in a dish and are an imperfect model system. So there are important questions about fidelity of organoids as a model system, meaning how well do brain organoid cultures model the in vivo brain? I think these can be separated into two general categories. And the first is, do brain organoids generate the cell types found in the in vivo brain? For these types of questions, the BICCN atlases are extremely useful because they provide a reference to which we can compare the cells generated in a dish from the organoids to the in vivo brain. There's also a second level question, though, which we also want to address. And that's, do brain organoids model the inter-individual variability of the participants from whom they were derived? The second question I think is critical because there's an assumption that the cells in a dish model the behavior of the cells in a human. So in order to investigate this question, we need to generate iPSCs from a really well-characterized population. So we did this through collaboration with the Infant Brain Imaging Study, which recruited families who already had a child with autism. And then the study longitudinally imaged the brain of the next child in the family from infancy at 6, 12, and 24 months of age using MRI. This allowed them to extract features of cortical size and specifically cortical surface area through development. We then generated induced pluripotent stem cells from 18 participants in the IBIS consortium and differentiated them into cortical organoids. So this represents a really unique resource for which to generate an iPSC population because it allows the assessment of in vivo to in vitro relationships, as well as the study of molecular mechanisms leading to autism. So we found that when sampling about 200,000 cells using single-cell RNA-seq across 2 different time points at both 14 and 84 days post-differentiation, that these in vitro models of brain did recapitulate the major in vivo cell classes of neurons, radioglia, intermediate progenitors, cycling progenitors, and also choroid plexus cells. This was done by comparison to data generated by the Kriegstein group as part of the BICCN. And note that we, like others making in vitro models, see poor subtype specification. So instead of seeing splatters, we see smudges. We are not getting a one-to-one mapping of an in vitro to in vivo relationship, and instead see these smears of mapping. Also, as expected, we're missing some cell types like vasculature and microglia that are differentiated from different germ layers. But overall, human cortical organoids are producing the expected cell types. So our next question is about, do organoid phenotypes relate to the cortical size of the participant from which they were derived? We first pursued this using a simplistic measurement of just organoid cross-sectional area as the organoid differentiates, and through some simple imaging methods. Organoid growth rates, interestingly, were nominally correlated with the growth rates in vivo of the cortical surface area. And this suggests that organoid development is actually predictive of inter-individual variation in cortical growth. So we next wanted to understand if specific genes or cell types within organoids were associated with cortical surface area of the participant from which they were derived. And we found, interestingly, that one subcluster, labeled here as subcluster 22, had over 3,000 genes that were correlated with infant cortical surface area growth rate. Those genes were enriched in Rho-GTPase and cell cycle processes and include known microcephaly-associated genes, as shown here. This suggests that not only can we use organoids to model inter-individual variation, but we can also learn more about the specific biological processes that are associated with differences in human brain size using organoids. We then used a pseudotime analysis to better understand the developmental trajectory of that subcluster that had so many genes associated with cortical size. And interestingly, that subcluster was right at the crux of the fate decision point between progenitors and neurons. And this suggests that gene expression changes during this critical fate decision between neurogenesis or self-renewal lead to inter-individual variability in the size of the cortex. And this is consistent with some previous theories about what determines differences in brain size, like the radial unit hypothesis. So that describes a previous study, and now we're expanding this IBIS iPSC resource to 115 participants, well above the 18 in the previous study. And this will allow us to better assess the fidelity of organoids for modeling brain growth differences. And it will also allow us to test for diagnostic differences between autism and neurotypical as well. So the next thing we're interested is in what causes the variability in phenotypes across individuals. As Evan talked about, there must be genetic variation, environmental variation, or their interaction that contributes to variability in phenotypes across humans. Genetic variants may lead to increased risk for disease or any other phenotype in the presence of some environmental stimulus, but maybe less so in the absence of that stimulus. So these type of gene by environment study designs have been difficult to pursue in epidemiological human genetic studies so far because of the difficulty of controlling and measuring environmental exposures in human populations. But in organoids as a model system or other cultured neural progenitor systems, we can control environmental exposures really, really well. So we pursued that using primary human neural progenitor cells from 80 different individuals, each of which have inherent genetic variation. To those, we added valproic acid, which is a known environmental risk factor for autism, at clinically relevant concentrations for 48 hours, 2 days, or vehicle. We then tried to identify how genetic variation alters gene expression and chromatin accessibility within each of these conditions. And then we conducted response QTL analyses to see if there's a significant difference of genetic variant effects within these different conditions. So we actually detected many QTLs through vehicle or VPA exposure, but importantly, we found that the effect of genetic variation was modulated by the presence of an environmental risk factor for autism. And all these ones in black circles here across the entire genome show significant gene by environment interactions. So this shows that genetics interacts with environment to alter molecular traits. We also have much higher power with only 80 samples here to detect gene by environment interactions in a dish than in human populations, likely because these environmental exposures are really well controlled. So these data might be useful in identifying individuals that may be more or less susceptible to environmental toxicants, as well as biological processes that could be used to prevent the negative effects of these environmental exposures. So we're now expanding this approach to use inherent genetic variation across a large population of iPSC-derived organoids to see how genetic variation affects response to environmental toxicants. So overall, we're using a unique population of iPSCs from well-characterized individuals to study the fidelity of organoids at both the cell type level and at the level of inter-individual variability, but also to understand the biological underpinnings of their variation, which can be used to study gene by environment interactions in a dish. So I'd like to thank all the people that have contributed to this, and thanks so much to Nenad and Evan for the invitation to speak here today.
Great, thank you so much. So I think all four of these presentations have brought up a lot of very thought-provoking questions, and hopefully for the panel, especially sort of setting the stage for the kinds of questions that we hope to address in the next 40 minutes. I see there is one question here.
This is Stefano Marenko speaking.
Sorry.
Hi,
okay,
Jay.
go ahead.
You use these organoids where you transform back, you remove all the epigenetic marks, right?
Right.
And then you have a model where you can study genetic variation independent of those epigenetic marks. However, you could also derive from the same cells, you can transform directly into neurons, maintaining the epigenetic marks, right? And then you would have a way of measuring the difference, the environment, the environmental effect, and you could tie it back to the history of these individuals. Would that be a possible strategy or not?
It's an interesting point. So you could use, like you're saying, transdifferentiation systems to go direct to neurons, which may not erase the epigenetic profile. But I would say that for most environmental toxicant exposures, the exposures are really poorly controlled. So they're really difficult to know and oftentimes not recorded well. So for example, if there was an exposure during fetal developments, you wouldn't really know that exactly well. Whereas if we reprogram them to iPSCs, we can sort of wipe out that epigenetic profile and then have a very controlled exposure level. So by doing that, I think we can sort of really tightly control the exposures and then have a better understanding of gene by environment interactions.
Thanks.
But my hypothesis, yeah, thanks.
Anqui.
Okay, great. I want to make two quick comments. The first one is a general one, which is that I really want to concur with the emphasis on studying spatial transcriptomics. As Evan mentioned, as Genevieve emphasized, and also as Mercedes emphasized, this is important not only for cross-species comparison, really understand what similarity and difference comes from, especially the proportion of different kinds of cells, and also, how the newly emerging cell types, for example, in the human, may derive from, other kind of cell types that you may find in mouse or other species. And also in terms of disease changes, things like that. Just wanted to emphasize that. And I want to use my second comment as an example to say why that is important. And that is related to the midbrain derived GABAergic neurons that are found in the thalamus. So it is well known that in the thalamus, there are several GABAergic neuron dominant nuclei. For example, ventral lateral genicular nucleus, the intergeniculate leaflet or IGL, that's right above ventral lateral geniculate nucleus, and also this subparafascicular nucleus. All those nuclei have a lot of GABAergic neurons, and they are all midbrain derived. So even in the mouse, the GABAergic neurons belong to the midbrain class. And then there are other GABAergic nuclei in thalamus, like the reticular nucleus, well known, zona incerta, both considered ventral nucleus of the ventral thalamus, and those neurons are derived from hypothalamus or kind of in between. So when we say that the midbrain derived GABAergic neurons are much more dominant in the human thalamus, I'm just wondering exactly where they come from and whether they are related to those well-known GABAergic nuclei, or now they are also penetrating the glutamatergic neuron dominant nuclei, such as dorsal LGN and many other of the excitatory nuclei. So this is kind of posing as a question to the speakers, Kim and others as well. But I just wanted to say that again, spatial information would provide a much more definitive answer in understanding the cross-species difference.
Thank you, Kangui. And I think that's a great segue also to just sort of a more general question, which is, how can these atlases help us to clarify some of these, human-specific differences that are being uncovered? And what are the really important features like spatial data that will really allow us to make these kinds of analyses, durable and clear?
Yeah, exactly. Clarify.
Kim,
that's why I would really want to support what you guys said. Yeah.
Yeah. Does Kim or Tom or Arnold, any of the panel members, want to weigh in on some of these questions?
I can weigh in from my perspective, but I'm sure also that others have a lot to add as well. So I think, as I sort of hinted at, we've had trouble just on the single cell lacking spatial data alone, mapping these to specific GABAergic subtypes. So figuring out sort of which specific. So it looks like they map to most closely to neurons in the superior colliculus. So, but there's which of these various mouse GABAergic thalamus subtypes they might map to. We don't have that yet. So maybe someone else knows a little bit more about that. But a lot of these subtypes seemed relatively similar to one another in our analysis. Yeah. So I think we need more information.
Phil?
Yeah, I mean, I had a sort of slightly different sort of comment, which was I think especially for what we're working on, which is, looking at these pathological relationships and trying to understand cell-cell communication and be related to that. As I mentioned, the cell states that we ended up prioritizing in our analysis represent less than 1% of the microglia or 1% of astrocytes. And so the spatial data is going to be really key to resolve these microenvironments and these cellular communities, because I think we're missing a lot of information because these rare pathological cell states are swapped out by all the other cells which are still in the normal state. And again, it depends on your condition of interest, of course. Glioma is going to be very different. There's this really extreme environment. But in the case of disease like Alzheimer's, which progress over decades, the number of pathologic cells is actually very small, and the brain is relatively well functioning at that time. So this topology is going to be really key. So we can zoom in and also related to the important features like aggregates of pertinopathy, because those cells are going to be qualitatively different than the ones that are farther away or near a blood vessel, et cetera.
Definitely think that's a key point. And we sort of have two threads going on spatial with regards to disease and also with regards to evolution. But I see Tom and Arnold here, so maybe they can offer some thoughts on the evolutionary side. We can come back to the disease questions too. Tom?
Yeah, so as many of you know, the development in humans is dramatically protracted, including, Mercedes has done beautiful work demonstrating that many interneuron types migrate postnatally for protracted periods of time. And so I think that looking at development will be absolutely essential to answering some of these long-standing questions about the origins that Hong-Kui was bringing up. And spatial data absolutely will reveal these, dramatic examples of cells migrating through the tissue environment that will be essential for understanding of what makes us human. But also, I think a really opportunity for the field to also use this information as a handle into cell therapies, so cell replacement therapies that are now becoming, really coming into trials thanks to the foundational work from Arnold and Arturo. You can basically learn about the migratory patterns of these cells, and as we bring these therapies into patients eventually as a field, I think this understanding of these migratory streams will be essential, critical. So I highly agree with Hong-Kui about the need for more spatial data across development.
Arnold?
I just wanted to make a comment. I think, everything we've heard is really terrific, but it underscores in many ways the importance of studying human brain development, at early stages. Most of these neurodevelopmental disorders, autism, schizophrenia, begin to manifest prior to birth, and in some of the cell types that are just, now becoming better understood, and especially progenitors that may be human or primate specific. And the problem is that in order to study them, organoids are wonderful functional models, but they really need to be based on ground truth. And for that, there need to be studies of primary tissue. And, the concern I have is that the NIH is no longer funding that type of fundamental work, and it's shifting to other parts of the world. An example is today, there was a nice paper in Science coming out of China that looked at the development of the ganglionic eminence and neurons that are expanding because of particular progenitors found in the human brain, not found in other animal models like mice, for example. And so I think that a lot of those fundamental ground truth studies are likely not to come out of the NIH anymore, but come from other labs in other parts of the world. And so I just, this is my comment. I think it's an unfortunate circumstance at this particular moment, especially as the excitement for organoids sort of highlights the importance of looking at lineages in human brain development, which are not really well understood yet. So that
Arnold, can I make a quick comment before you, Tom? Arnold, I agree with you 100%. At least Brain is not abandoning the work you all are doing, so just keep doing what you're doing. I think that to the extent that other labs and other countries are investing in this work, as long as we're able to see each other's data and incorporate it into a larger knowledge base, that's all to the good. So let's just focus on what we're doing, and we will continue to fund the best science out there. That is my commitment as long as I'm around. Okay.
Thank you, John, and thank you for everything that you're doing for the community. This is really important. I just wanted to bring back and, maybe highlight some other opportunities. I think, with the Connects program of the Brain Initiative, I think an unmet need is also to map and contextualize these cell types in the context of connectivity. So I think there are new emerging tools that will allow us to do that, both utilizing primary tissue cultures, organoids, as well as other methods. So the extent to which we as a field can start working together on deploying better methods, and I think it speaks to something that Philip also brought up, because I think tools that allow you to map synaptic connectivity. Also, we should not forget about glial connectivity and the interactions between neuronal and non-neuronal cells. I think we need better tools, and we are poised as a field to begin to venture into that direction in the next 5 years in a systematic and coherent manner that I think will also answer some of these long-standing questions.
I think Steve McCarroll, another panel member, had a quick point. He can't seem to raise his hand, but then we'll get to Phil after.
Yeah, so I'm excited to start to see more spatial information and spatial studies in more diseases. I have to say, looking at some of the early things that have come out, I find myself yearning for a couple of things in that literature. One is publish negative results as well as positive results. Because I think when you're in the early days, like doing kind of the first spatial transcriptomics or whatever analysis of a new disease, the negative results are just as interesting as the positive results. And a field may have some enormous number of like theories and hypotheses about cell-cell interactions and things like that. And it advances the ball to say which things actually weren't supported by the data, right? And if you don't do that, then those questions just never get settled, right? And then second, I also think it's really important to acknowledge when like the picture that's emerged at the end of this study isn't necessarily a spatial one, right? Because in particular, like in some diseases, it's just going to be a cell autonomous process. And in other diseases, it's just going to be a system-wide process that's the same, that's due to some circulating factor or something that's the same everywhere. And then there will be other things at this intermediate spatial stage where there will be focal findings or lesions or plaques or whatever that organize the biology of the space around them. And I think it's really important to just think clearly and say at the end of a paper what that emerging picture is, and whether or not a spatial technology was used.
Phil.
Yeah, thanks. I want to go back to Arnold's point, I think, in terms of studying the younger brain, the developing brain, and the early adult one. The data are really quite sparse here. And so I think this is important both because we're beginning to realize that the source of a lot of the degenerative diseases are actually found in development. And certainly things like resilience are going to be established there. And there's the effect of education, of course, which is protective for a lot of these diseases. These are not molecularly understood, and we're going to have a hard time if we don't really understand how the brain develops and what variation is achieved, by the time our brains are mature, right in the mid-20s. And so I think that sampling of especially the older younger adults, I think is really sort of lacking at this point, but also, of course, more data all along the way. And I think the other thing is also that, especially the studies that we do, tend to use, of course, older individuals. So we're looking at brains that are 70 to 110 years old. We're trying to infer what like a healthy, cell state is, but we don't. But this is based on the collection of samples that we have on hand, and so we don't really have great references. And so, having a better sample of the diversity of cell states in younger people, sort of in midlife, is going to be essential. And I think we actually have a project now to sort of begin to address this question, but we need a lot more data from these younger and middle-aged individuals to really understand, the aging trajectory.
And I'll just say this has been a central focus of many of the projects in the BICAN consortium. So it's great to hear you say that. And I think this particular group is very passionate about getting that data to the community for all of these reasons that you just described. Mercedes.
I just want to follow up, and I think this is also an opportunity for the community to think about how we can do more outreach. I think that gap that Philip was bringing up, those are pretty rare tissues to get. And so I think the more we can let the public know about what we're doing, I think it's exciting to bring in disease because that additionally gets their public interest. And that would allow for more conversations about, those tough, challenging conversations, but how do we interrogate the human brain and get that neurotypical sample that is rare, but we want, and I think is really important because so much of what we're looking at is a baseline going to have a comorbidity, have some, even our surgical resection sections, they're not normal tissue. And so the more we can get neurotypical tissue, I think is also going to be critical to see the, what's our true ground truth, not just in disease, but in development.
If I may add to the last two comments, and I think Tom's, thank you. One also thing that we have to keep in mind that even though there is a tremendous amount of conservation, evolution works by tinkering rather than inventing new things. So pretty much whatever we found in humans will be able to be modeled, at least at the level of cellular molecular in some other species or models, stuff like that. But very important thing that to keep in mind that even when things are conserved, they are happening at different time and different context. And to me was one of the biggest obstacles to understand human biology is understand the second half of the prenatal development, because if you think that part of the development happens in utero in humans and primates, but in rodents happens outside, they're already born basically. So it's equivalent to mid-fetal, like somewhere in the middle. And so, they're already exposed to different environments. So even if you can model that in other species, it's very hard to model that context, because most of things in second half happens in rodents postnatally, and they're already exposed to environmental and other stuff. So I think that brings to what extent we should really increase the use of non-human primates, because that part of the development still happens in utero. And plus we have all other geometrical differences in the size and complexity and many other things.
Partha, you want to go ahead?
Oh, yeah, thanks. Hi. Yeah, I just want to reiterate the point that has been made now a couple of times, but because this is a data analysis, future data analysis focused workshop, there's a great opportunity for crosstalk between BICAN and Brain Connects. And in fact, I wish we knew about the striatum parcellation earlier a little bit. We could have maybe even joined in there. But generally, the Brain Connects in primates is trying to make kind of regional mesoscale projection maps. And to what extent does that correlate? And I'm talking primates, mouse, we have studied this, but in the marmoset, the macaque, and in the human, to what extent do those sort of regional mesoscale projection maps correlate with the spatial omics data? I think it's a real opportunity that has a lot of legs, and it's a good opportunity for these two consortia to work together in the future, I think.
Tom?
Yeah, just quickly wanted to also echo some of the things that Jason brought up in his presentation about inter-individual variation. I think one of the opportunities for BICAN is to also capitalize on the NIH investments in the whole genome sequencing of individuals, and particularly the assembly of the pangenome, because if we want to understand the inter-individual variation, I think a lot of our reads historically have been mapped onto AG38 or some of those more reductionist genome assemblies. But now that we have T2T sequencing of many individuals in the pangenome, I think one of low-hanging fruit opportunities to actually go back to the sequencing reads that we've so painstakingly generated, and actually look back at, what have we missed? What are the missing pieces? What can we learn from that? So I think there is a lot that we as a community can do.
Mark, go ahead.
Sure. So I was really heartened to hear the interest in inter-individual variability, and I think that's a great thing, obviously, to look at, particularly in relation to disease. And I'd just like to say that, obviously I've looked at this a lot, and it's surprising how many samples you'll find where there's just RNA-seq and there isn't even a, genome sequencing or gene chip done. And I'd just really like to sort of give a plug that every time one takes one of these precious samples and does the RNA-seq, it'd be really nice to have just even a simple gene chip that's well registered to it, because I think it really makes for a much more valuable data set. And the second thing I want to say is that, there's a lot of discussion about cell proportions and things like that. And these are very interesting to look at, but they're very, very sensitive to, a lot of the ways these experiments are processed and how the, things are filtered and so forth. And I think it's very important to preserve that, additional information so one can accurately think about the cell counts and so forth.
I'd like to just jump in on that, because I think that's a really key point, and something that we've been realizing as we've been scaling up the number of donors that we've been running slide tags on in the atlasing efforts that we described, is that once you actually are able to delineate brain areas and really control for boundaries, the proportions of cell types become far less variable. And so we think one of the key challenges that we've had with these single nucleus RNA-seq data sets, especially for glia, where it's very hard to know where you are in a structure, is just the inability to really parselate these boundaries and the variants that require such large sample sizes to get beyond.
We have Joe, then Marga Behrens, and then Dirk Keene.
Probably you can't hear me.
Now we can hear you.
Oh,
Yeah.
hey,
We can hear you.
I just wanted to echo something that Tom said about, there is a lot of genome-wide association study data that can be put into a single cell context. So like Mike Snyder had a paper in Nature Biotech last year that demonstrated it pretty well, that you can actually, rather than, I mean, it's great to have a lot of single cell profiles from individuals, and that'll take a while, but you can already take the population data and use it to predict risk scores for what cell types are most vulnerable in those cases. So I think they demonstrated 3 or 4 examples of that, and it was really powerful. So I think to again, that what Mark said is that, yeah, I mean, you need to, if you're going to do RNA-seq, you should be doing in parallel multiome, so you can get that kind of information that you're going to need to identify those variants.
Marge, you are next. Thank you, Joe. Very good point.
One of the things that I wanted to bring up is the importance of setting up a way of obtaining samples from newborn to adolescence. We are really lacking those samples, and I think it is extremely important to understand the development in childhood, pre-adolescence, what is happening in the brain. And unfortunately, I don't know the rest of the whole BICAN here, but availability of such samples is really lacking. And so how can we do to expand such that we can map those brains, as well as we have done the prenatal brain and the adult brain?
I could not agree more with you, Mark. I mean, that's one of the critical, problems in developmental studies. And as I always make a joke, first thing you realize about me that I have heavy accent because my brain was in another country when I was a
yes.
child.
Mine too.
Yes, and it tells you the reason I want to go back to my previous comment, and it's not genetic, it's just not being raised. And it tells you the power both of environment shaping the genes at that time and expression or circuit development. And we have absolutely very little. No, and you are absolutely spot on. I mean, the problem is getting high quality brains, and the problems are really related. As you could understand, it's not an easy and stuff. So I do think that maybe something that can be done at NIH to emphasize this as a future focus of I think will be important, and I will be happy to participate, help, and brainstorm about that. But I think that's a critical point for any kind of developmental studies, because what we have now is a lot of mid-fetal development, not a lot, I mean more than, and then just basically adult. And there is a whole this window that is essential to understand all neurodevelopmental disorders and susceptibility. So completely one of the key points. Thank you.
It's a perfect time to turn to Dirk.
Hey, everybody. I'm really inspired by Steve and Phil and Mark and others. Knowing as much as we can about these different samples just makes total sense, right? We should do that, but we have to think of who these people are that are donating their samples and how much do we know about them. And so to Steve's point, publishing negative data, I'm surprised when we publish our papers at how little that we're really talking about the people who've donated the tissues that we're studying. One of the most, I think, astounding things that I've learned as part of BICAN is how hard it is to actually come up with anything neurotypical, right? I don't even know what that means. The vast majority of the young adults that we have been able to procure have some sort of an accidental or sometimes intentional overdose. A lot of people would argue that those are definitely not neurotypical. Older people have vascular pathology. We see Alzheimer's pathology in a lot of our 40-ish-year-olds. A lot of these studies don't even look for Alzheimer's disease pathology in 40 and 50-year-olds. And so for disease, Phil is studying the ROS-MAP cohort. It's one of the best characterized cohorts of people. The more we can learn about these people during life, longitudinally, both from a cognitive perspective, a behavioral perspective, imaging, biofluids, genetics, the more likely we're going to be able to connect what we're learning from a pathology perspective, and most importantly, from all of these really cutting edge single cell and spatial methods to link back to these diseases and these people, and be able to first and foremost develop biomarkers to understand what's happening when, especially early, and then to, you know, develop experimental models that are accurate, that we can actually test relevant hypotheses in a relevant system where we'll get relevant results. And so, you know, my pitch is we really need to be aware as a community that the people donating their brains in a lot of these studies are just not, you know, what is normal. And the more we know about these people, especially when we're talking about low numbers, you know, the more we're going to be able to interpret some of the different outliers and things that we see. And this also highlights how important it is for studies like Evan and Steve's UM1, Phil's, you know, large scale studies in the ROSMAP cohort to get numbers. But the other thing to remember is, as pathologists, when we're characterizing these brains, typically we're looking at a handful of brain regions, many of which are totally irrelevant to whatever cognitive function or other things are happening in these brains. And so we need to understand the neuropathological assessments are pretty basic, and we need to be thinking about how to develop those. There was a question about digital pathology, and there's huge potential in quantitative pathology to help inform how we understand what's happening with these assays. And then, you know, let's look early. Let's find ways to look early in these disorders, or even in people who have advanced disease. Look in their brain, in areas of their brain that are less affected, so that we can really find the early parts of this process, which is going to enable us to diagnose it and treat it effectively. And so anyway, the cohorts we're using are great. It's really, really hard, to Marga's point, it's really hard to get really great specimens. And, you know, first and foremost, they have to be high quality. But we really need to have well-characterized people too, especially when we're transitioning into disease. And the cohorts that we're referencing, our reference atlases, really need to be apples to apples when we're starting to study our disease, whether that's age, sex, genetics, exposures, you name it.
Can I just say something in support of what Dirk just said? Because we've actually looked at it quite extensively in our project, because the question came up about including people who have kind of more complicated histories, maybe, you know, maybe some drug use, or at the end of life, or suicide, or something like that. So we looked really carefully at the question of what do you get and what do you lose by defining controls narrowly and rigidly? And the answer is you lose tons of power and you gain nothing in terms of signal to noise. So and hopefully we'll be able to clarify this in the preprint. I think it will be helpful in disease studies for people to be able to think, to know that it's okay to think expansively about who you use as controls.
We have Sarah and then Mark, and we have 8 minutes left.
Thank you. Yeah, so I'm really enjoying this conversation around variability of different types. And I just want to mention, you know, for Mark's point on the genetics, I mean, from the sequencing based, you know, mostly suspension cell technologies, you obviously get some coarse level of genotyping, particularly from the multiome, from the data itself of its sequencing. But the other points about kind of lifespan variability, and particularly sort of neonatal, pediatric, and then early adulthood, and then the sort of other side of it is just aging. And there's, you know, on top of that, I also want to, you know, to Marga's point, I also want to mention pregnancy. I mean, there's, you know, beautiful work from Fiona Dutch that shows neural stem cells changing during pregnancy in mice. Is that the same in human? We don't know. It's obviously really tricky to access samples, but it will be absolutely fascinating. I mean, for other tissues like the gut, it's much easier because you can kind of biopsy and so on. I don't work in the brain, and I'm you know, not, or at least not mainly in the brain. And so I've got more knowledge about tissue acquisition for other organs, but I've spent several hours today. So this is my evening. I'm in Cambridge, UK. I've spent several hours today talking about tissue access and, you know, access to organs, either from deceased transplant donors or from postmortem tissues, or are there other new ways of maintaining fresh tissue, like putting cadavers on perfusion, and that kind of thing? And, I wonder, I would be curious, just a question to this community. I thought this was more about data, but I'm just curious about tissue access. Is kind of freshness an issue for the brain? Because the GTEx data kind of shows that brain is very stable, where some of the internal organs sort of degrade quickly. I mean, I would just be curious about the consensus, and also the kind of sources in the US and here in terms of tissue access and feelings about quality of tissue and freshness and so on, and whether it's an issue at all.
I think this is Nenad. I think that's a very important topic. How fresh, that also depends extremely on ante-mortem and post-mortem conditions. So that's why PMI is not a good correlate of the quality of the data. It's just because, fortunately, people die from different circumstances at different time and at different. So that's another factor and going back to what many have said, the more we know about these samples, not just their life history, but also ante-mortem and post-mortem condition, the more we will know. As we know, PMI affects certain transcripts differently, certain cell differently, everything, all of that, we'll get a better pictures with more samples. That's, I think, the only way forward, to collect more, but have a very good record of what we are collecting. I think that's a critical.
So what is the ideal? What is the dream kind of biochemically most fresh, like from a clinical, surgical, resection
surgical
material.
tissue,
Yeah,
right?
yeah,
The
yeah,
ideal,
yeah.
the ideal
For
is
sure,
you get, right? Because a lot of the assays that are being done really require functional cells, and that doesn't last very long. But it depends on what you're looking for, right? If you're an electrophysiologist, then you're not going to be able to use postmortem tissue almost certainly. If you're studying, various proteomics, then it might be a little bit more. Your assay might be a little bit more robust in older postmortem tissue. And so I agree completely with Nenad that perimortem conditions are really, really important. Knowing more about how that person died and how quickly they died and what happened to them right after they died. It matters if a person died outside or inside here in Seattle. If they died outside, their brain ends up being cool, and it actually matters. So it just depends on the assay and what you're looking for. And then you have to have quality metrics that are going to tell you what that looks like.
And just really to make a comment, because Sarah made a very precise comment, I just want to, the GTEx brains were exceptionally well done because they put basically a cold mask and ice on the head immediately after. So that's due to the people who were collecting that. So they really proactively thought about it. And that was an example of extremely well designed project colleagues. And to do this, we PIs can do very little. We have to be engaged more with people that are collecting tissue, with medical examiners, neuropathologists, and stuff like that. That, I think, is the number one issue here. To get better quality tissue, we need to connect better scientists with people that are really doing this. And so they have to think ahead of time how to improve the quality. And that's why GTEx was actually done well.
Yeah, I mean, I'm in a clinical school now, professor of a clinical school for the first time. And so this is kind of a topic that's easier for me to tackle now. And as I said, I spent time with transplant surgeon, intensive care clinician, and actually scientific kind of coordinator 3 hours a day. So it's kind of fresh in my mind. Yeah. Anyway, thanks. Great conversation. Thank you, everybody.
We have a few more minutes. Mark?
Yeah, I just wanted to quickly say, it's sort of related to what everyone had, Sarah and Nenad and others saying. I do think GTEx is a great thing. And in particular, I wanted to really mention this thing called DGTEx, which I think is a really nice project that's really going to look at development and fill in these developmental gaps. And one of the, I think, key things in this project, which I think is important, is obviously we're talking about the brain today, but the whole point of DGTEx is to look at the whole body and to really look at all the different tissue samples in relation to each other. And I really do think that's a valuable thing to do, and not just to look at the brain. In any case, I just wanted to mention that briefly.
And Mercedes, I think you are the probably last person. So
Yeah, I just kind of want to follow up on what you were saying, Nenad, like that to fill in that gap window, what I think ranges from the third trimester all the way through young adulthood. I think we'll be working with clinicians, especially kind of neonatologists, pediatric. Here at UCSF, Betsy Crouch from neonatology has really tried to create like a conversations with the fellows so that there can be more contact between the bench scientists and the clinicians to have those discussions. Because I think that's a big difference in trying to get these younger gap ages is those conversations in some ways have to happen in a much more sensitive fashion because there isn't that time for preparation often. But it's critical, as I think everyone's highlighted, with PMIs, comorbidities, and are even those hospital cases ones that we'd want, given that they're probably intrinsically already have other conditions that are going to influence the quality and nature of the tissue. But I think it's something that as a community, we really need to think about if we want to fill in this time period, both in, again, in whatever neurotypical, however we define it, but also in disease states at these critical ages.
Great. Any last comments from you, Evan or Nenad?
No, just thank all the panelists. I think we had a great discussion and speakers for putting the time into setting the stage. It was a really fruitful discussion and we covered a lot of topics.
And I had many questions, but I think it was important for others to ask anyway. So, and thank all our speakers for wonderful and informative talks.
Absolutely. And of course, we have the chat where the conversation can continue for the remainder of the meeting. But thanks again to the panelists and to you, Evan and Nenad, for moderating and for being on time.